Earth's living surface begins with a thin architecture repeated inside every breathing cell. That architecture is the phospholipid membrane, a skin of countless small, restless molecules. Each phospholipid carries a water-loving head and two water-shy tails. When water surrounds them, they arrange themselves with quiet physical certainty. Their heads face outward, while their tails hide together inside, away from water. From this simple gathering, flexible walls arise around living cells and organelles. Bacteria, leaves, eggs, and human tissues all depend on this boundary daily. Across the planet, phospholipids are renewed, broken, and rebuilt every living moment. Yet the same molecules can become a stubborn puzzle outside the cell or factory. In freshly crushed seeds, phospholipids swell into gums that cloud valuable vegetable oils. In rich egg yolk, they hold oil and water in a lasting embrace. In injured tissues, they can hide chemical messages within delicate molecular seams. For early organic chemists, these materials were beautiful but difficult to open cleanly. Heat and strong reagents could break them, but often with little grace. Nature, long before laboratories, had already found a gentler molecular key. That key was phospholipase, not one enzyme, but a careful, varied family. Some members live in digestion, helping animals manage the ancient language of fat. Others appear in leafy seeds, microbes, venoms, and the chemistry of inflammation. All recognize phospholipids, then choose one precise bond to loosen. The documented story begins with egg yolk glowing in 19th-century glassware. In 1845, Théodore Gobley isolated lecithin from the yellow yolk. He later found the same substance in brain, blood, bile, and seeds. Gobley showed that a fat-like material could carry phosphorus and nitrogen. Lecithin became a new living category, neither simple oil nor mineral salt. But its behavior remained deeply mysterious, especially where living matter met water. Near the century's end, snake venoms brought a darker biochemical clue. Physicians watched certain venoms damage red blood cells with frightening efficiency. When lecithin entered these experiments, the reaction became easier to carefully trace. Venom was changing lecithin into a more disruptive form of the molecule. In the early 1900s, fresh pancreatic extracts revealed a similar hidden power. Charles Delezenne and colleagues connected digestion, bile, lecithin, and cell rupture. The altered product became known as lysolecithin, a sharper membrane disruptor. A biological cutter, then called lecithinase, had stepped into scientific view. Through the 1930s and 1940s, purified enzymes patiently sharpened the picture. Wartime bacteriology identified a deadly bacterial toxin as Lecithinase C. By mid-century, biochemists increasingly divided phospholipases by the bond each one chose. Phospholipase A clipped one fatty tail from the molecule. Phospholipase C separated the head group from the oily body. Phospholipase D reshaped the head end, trading one partner for another. These names became a map for work too small to see. Researchers found phospholipase A2 especially abundant in pancreas and snake venom. In digestion, it helped turn dietary phospholipids into absorbable pieces. In venom, the same precision could become pain and injury. The enzyme itself had no malice, only fit, motion, and chemistry. At the microscopic scale, phospholipase settles beside a membrane surface. It senses charged heads above and crowded oily tails below. A water molecule is guided toward one vulnerable chemical seam. Then a single bond relaxes and the phospholipid changes character. One fragment may become a signal, calling nearby cells to respond. Another fragment may help fats travel through watery places. This is chemistry performed softly within crowded living neighborhoods. Modern industry noticed because the same softness could replace rougher treatments. As global vegetable oil production expanded, phospholipid gums became costly impurities. They trapped water, carried tiny trace minerals, and made clear oil harder to obtain. In the 1990s, enzymatic degumming moved from trials into edible industrial refining. First inspired by digestive phospholipase, engineers later welcomed microbial enzyme forms. Phospholipase changed troublesome gums into forms easier to separate. The oil cleared, while gentler conditions reduced avoidable material waste. In food kitchens, phospholipase also transformed lecithin-rich egg yolk. Modified yolk could help sauces, dressings, and fillings remain stable for longer. Bakers found its quiet touch useful for dough strength and crumb softness in bread. Dairy makers could guide flavor and texture through careful lipid changes. In soy lecithin, it helped tailor emulsifiers for chocolate and spreads. In biotechnology, related enzymes prepared phospholipids for research and specialized medical materials. In medicine, phospholipases became signals to study inflammation and cell communication. Their history reminds us that one enzyme can carry many meanings. A digestive helper, a venom weapon, a laboratory clue, an industrial tool. Across these roles, the common theme is practical, selective, lower waste change. Instead of forcing matter, phospholipase invites a carefully chosen bond to open. At life's oil-water border, greener chemistry may keep finding its future for industry. Enzymes Bio is run by people who believe enzymes are the future of clean industry. So we build one platform for startups, labs, and makers to source them without the usual hassle. Gentle on the planet, built for what's next. Visit enzymes.bio.